So we are going to see the cell junctions in this video. Before going into the proper topic, let's look at what you mean by cell polarity. So basically this is a cell, these are individual cells and they are resting on, these are epithelial cells and they are resting on a basement membrane, and this will be the region of that particular cell which will be opening into a particular, which will be related to a particular lumen or the external surface. Anyway, this surface over here of a particular cell is the apical domain, and the region which is in relation with the basement membrane will be known as basal domain. In between them, all the regions of the cell which is present on the lateral aspect will be known as lateral domain. So with this, we will move on to see the cell junctions. First, we will look at the classification of the cell junctions. So they are mainly of two types, unspecialized and specialized ones. As the name indicates, unspecialized cell junctions will not have any individual or unique properties. We will see that later in the coming slides. In the specialized cell junctions, we will have anchoring junctions, occluding junctions and communicating junctions. And within the anchoring junctions, we will have adhesion spots, adhesion belt, adhesive strips, hemidesmosomes and focal spots. We will see all these details in the coming slides. First, we will start with the unspecialized contacts. Then we will move on towards the different type of anchoring junctions, which is over these 5 types of anchor junctions. Then we will look at the occluding junction. Lastly, we will move on to see the communicating junctions. The first one, the unspecialized junctions. So this is the basic structure of a unspecialized junction. As the name indicates, I told you already, that is not having any unique properties. So these two layers over here is the bilayered cell membrane, and this is the cell membrane of one particular cell, and this is the cell membrane of adjacent cell. In between the cell membranes, we have certain proteins connecting between the cell membranes, and this will be known as CAMs, or otherwise known as cell adhesion molecules. And these molecules will fill up this intercellular area. And the other end of the cell adhesion molecules, for example, one end will be projected into the intercellular area, other end will be projected into the cytoplasm of the particular cell. And what are these fibers? They are the fibers of cytoskeleton. Examples, actin or microtubules or intermediate filaments. All these are types of cytoskeleton. So they will be connected to different types of fibers of cytoskeleton through this linking protein. So here it is labeled as intermediate protein, so you can call it as linking protein. So that is the basic structure of unspecialized contacts and the basic structure of cell adhesion molecules. Now, there are basic types of cell adhesion molecules, that is calcium dependent and calcium independent. Among the calcium dependent CAMs, you will have cadherens, and that is of various types. We will deal with multiple types of cadherens in this video itself. Then we have selectins, we have integrins. In calcium independent group, you have NCAM and ICAM, that is neural cell adhesion molecule and intercellular adhesion molecule. And these cadherens, they are mostly present in the epithelial cells. The selectins and integrins, they will be present in mostly in white blood cells or leukocytes. And we will see one example in the next slide of the selectin and integrins. And we will deal with multiple cadherens in the upcoming slides. This is an example of where the selectins and integrins are present in case of white blood cells. This is a white blood cell, characteristic white blood cell. And this is one of the glycoprotein present in the surface of the WBC. This is one of the integrin. So on the surface of the endothelium, we will have P-selectin and E-selectin. Basically, this is the lumen of the blood vessel. This is the lumen of the blood vessel, and white blood cells are present in the lumen of blood vessels. So, this lining cells will be the endothelium. Within the endothelium, you will have P-selectin and E-selectin. So, they will receive this glycoprotein present in the surface of WBC. And further, because of this adhesion, they will activate these integrins, and once the integrins are activated, they will be attached to the integrin ligands over here. So, now it is denoted as stable adhesion, that is shown over here. For stable adhesion, you need selectins, integrins both, and once the adhesion is completed, then the WBC will migrate through the endothelium into the tissue. So, this is one example where integrins and selectins play an important role in the immunity. Before going into further topic on the specialized cell junctions, you have to know what are these terms are meaning. One is homophilic interactions, other one is heterophilic interactions. Homophilic means, as the name indicates, so we already know between two cell membranes there are cell adhesion molecules from each cell membrane, and they will be joining at the intercellular area. So, in case of homophilic interaction, the cell adhesion molecules from each side will be of the same nature. You can see the same nature of cell adhesion molecules over here. Here also you can see the same nature of cell adhesion molecule. But what happens in heterophilic interactions? Here the cell adhesion molecule from one side will be different from that of the other one. Examples are shown over here. So, that means that is the meaning of homophilic and heterophilic interactions. In the coming slides, you will see multiple homophilic and heterophilic interactions. Moving on, this is a schematic diagram showing the specialized cell junctions. So, basically this one is a cell, this is another cell, this is the area of another cell. 3 cells are shown over here, and the intracellular components has been removed to show the cell membranes, interior of the cell membranes, or we can say exterior of the cell membranes, and the domains very clearly. We have removed the cell components other than the cell membrane. So, in this diagram, this part, the upper part will be the apical domain. The lower part, which is located over the basement membrane over here, that is the basal domain, and the sides will be the lateral domain. So, basically what is dividing into these particular domains? We can clearly say basement membrane related part of the cell is basal domain, that is clear, but what is separating between lateral domain and apical domain, that is this particular type of cell junction, that is the occluding junction or ZONULA occludens. So, that is the region of the cell that is more towards the apex when considering the zona occludens will be the apical domain, and towards the basement membrane this area will be lateral domain. That is the importance of ZONULA occludens. We will see the structure later. Then what is this? We can see ZONULA adherens, just according to this diagram we can say just beneath or inferior to the ZONULA occludens. That is not a correct description. Inferior or beneath is not applied over here, but still for this description purpose we can use the term. Then in between the cells you can see multiple gap junctions on the lateral domains. Then in the basal domain we can see focal adhesions, small focal adhesions are there. Then multiple hemidesmosomes are there. And look at here, you can see a very strong adhesion in the lateral domains of adjacent cells, and that is maculae adherens or desmosomes. That is also a type of anchoring junction. So, this diagram will roughly give an idea how they are located and where they are located, multiple types of specialized junctions. So, we will move on to see the details of individual junctions. First, we will look at the anchoring junction. The first one, the macula adherens or desmosomes, or otherwise known as adhesion spots. So this is basically a simplified schematic structure of the desmosomes. We have seen the location of these desmosomes in the lateral domain of a particular cell in the previous diagram. So let's analyze this schematic diagram. This is the cell membrane, and this is the cell membrane of the adjacent cell. We can see the cut through the cell membrane, and these yellow structures over here are the cell adhesion molecules present over here, and they are belonging to the cadherin group of CAMs. Here in previous slides itself I mentioned there are different types of cadherins, and the cadherins that are present in desmosomes are known as desmocolin and desmoglein. And they are attached to these fibers of the fibrous skeleton of a cell through an intercellular attachment plaque, which contains mainly two proteins, that is desmoplackins and placoglobins. That is not labeled in the diagram, but still this is the intercellular attachment plate. Components are desmoplakin and placoglobin. Then what are these type of fibrous skeleton component? So remember, for each type of cell junction there will be specific fibrous skeleton component that is attached to it. So in this case, in the case of desmosomes, this fibrous skeleton component will be intermediate filaments. These desmosomes are the most common type of cell junction, and these desmosomes together, this desmocolin and desmoglein, they together form a zipper like arrangement, that is forming the continuous cadherin zipper. Now we will move on to see the next type of cell junction. The next one will be the ZONULA adherens or adhesive belt. We have seen that in the first schematic diagram, just below the ZONULA occludens. And we will analyze this diagram. These are the adjacent cell membranes similar to the previous diagram, and in between the cell membranes we have again cadherin group of cell adhesion molecules. We can see calcium has been shown over here. So basically that means they are calcium dependent cell adhesion molecules. Basically cadherins are calcium dependent, and these molecules, as we can see, they are homophilic interaction, and that will form a lateral adhesion between epithelial cells, and they will provide mechanical stability. So as we saw in the first diagram of the cell, they form a continuous band which passes all around the apical part of the epithelial cell. So this was the previous diagram. So here is the ZONULA adherens, and which will be passing continuously all around the apical part of the epithelial cells. And as you have mentioned earlier, the cell adhesion molecules present here are E-cadherins. On further detailing, we can see the E-cadherins over here, and over the cytoplasmic side there is a protein, a component is known as catenin, and the catenin will be further related to or attached to another protein, that is vinculin, and to this vinculin the actin filaments over here, which are part of fibrous skeleton of the heart, will be attached, and these actin filaments will be attaching to this vinculin with the help of this particular molecule that is shown in blue color in this schematic diagram, that is the alpha actinin. So this E-cadherin catenin complex, which will binds to vinculin and alpha actinin, and that is required for the interaction of these cadherins with the actin filaments of the cytoskeleton. And this complex of all these structures will function as a master molecule in regulating a lot of functions. Some of them are cell adhesion, some of them determines polarity, some of them will be related to differentiation of the cell, and cell properties such as migration, proliferation, survival of the cells. All this will be related to this ZONULA adherens or the adhesive belts. The next group of anchoring junction will be the fascia adherents or adhesive strips. So basically, before zonula adherents was a continuous structure all around the cell, but in this case of fascia adherents, these areas of attachment are in the form of short strips. They do not go continuously all around the cell, and the location of this fascia adherents is also different. That is mainly present in non-epithelial cells such as smooth muscle, intercalated discs of cardiac muscle, and in junctions between glial cells and nerves. The structure is almost similar to that of zonula adherents, and it will also contains ZONULA occludens 1 protein found in tight junctions, that is ZO1 protein. The next group of anchoring junction is hemidesmosomes, that is shown over here. These are the hemidesmosomes. In the first diagram of the cell, we have seen the location of the hemidesmosomes, which is related to the basal domain of a particular cell. And look at this. Where is this located? They are located in epithelial cells which require strong stable adhesion to the connective tissue lying beneath it. So, for example, cornea, skin, or mucosa of the oral cavity, esophagus, vagina, etc. And we can see the structures over here. This is the intracellular side, and this intracellular attachment plaque, this protein, link protein, which contains multiple types of components. One of them will be plectin. You can see the labeling over here. This is the plectin. So, it integrates all elements of the cytoskeleton. Next component will be BP230, and that is also known as bullous pemphigoid antigen 1, which is shown over here. And the another one will be erbin, and this is important in associating BP230 with the integrins, and all these structures forming the intracellular attachment plaque in case of hemidesmosomes. Now, earlier we have seen the desmosomes and all. Don't confuse this with the desmosomes, because in desmosomes the cell adhesion molecule that is present over here in desmosomes, that was cadherins. But in this case of hemidesmosomes, these CAMs or cell adhesion molecules belong to integrin family of matrix receptors. And what are these individual components of this cell adhesion molecules that is present over here? We can see multiple components over here. One of them will be alpha 6 beta 4 integrins, and it's extra. You can see the integrins over here in somewhat yellow color, and the extracellular domain of these integrins, that is over here, which will enter into the basal lamina. This is the basal lamina in this diagram, and it interacts with type 4 collagen, and this type 4 collagen will contain laminins, laminin 332, that is shown over here. So, this is the laminin, and that is the association of alpha 6 beta 4 integrin with the laminin 332, and thus they form a structure. This association will form a structure that is known as anchoring filaments. Anchoring filaments. We can see the anchoring filaments over here. The next group of cell adhesion molecules, next component of this cell adhesion molecules in case of hemidesmosomes include type 17 collagen, that is shown in somewhat red color over here, and another group one will be, that is not shown in the diagram, that is CD151. What happens if any of these components will get deranged or deformed? Such conditions include one of this prominent or very clinical condition, that is bullous pemphigoid. The next type of anchoring junction will be focal adhesion, or otherwise known as focal spots or focal contacts. These type of adhesions or anchoring junctions, we have seen this in the basal domain of a particular cell mainly. So, it creates a dynamic link between the actin cytoskeleton and the extracellular matrix protein. In case of hemidesmosomes, the cytoskeletal component was intermediate filaments, you would have noticed in the diagram. But in case of focal adhesion, it is again actin filaments. And the cell adhesion molecules which are present over here, they belong to integrins group of family. And these integrins, they include actin on the cytoplasmic side of this cell adhesion molecules over here. They include actin binding proteins and regulatory proteins, example talin, paxillin, vinculin, etc. And the next component of this integrin group of family will be the transmembrane connecting region, which we can see clearly traversing through the cell membrane. The extracellular phase includes of this particular integrins include extracellular matrix glycoproteins, and usually they will be laminin and fibronectin. So, these focal adhesions, they play a very important role in sensing and transmitting signals from the extracellular environment into the interior of the cell. So, they will be able to detect contractile forces or mechanical changes in the extracellular matrix, and through this integrins they will be converted into the biochemical signals within the cell. So, this property which is exhibited by this focal adhesion or focal contacts is known as mechanosensitivity. One interesting point to note here at this point is we have mentioned about anchoring filaments and we have seen anchoring fibrils. They are not the same, I have mentioned it earlier. Anchoring filaments will be roughly corresponding with the one of the anchoring junction, and anchoring fibrils is just a component of one of the anchoring junction, and anchoring fibrils we have seen in relation with the hemidesmosomes over here. So, this is a diagram showing the two types of anchoring junction present in relation with the basal domain of a cell, and this is the structure of hemidesmosomes, which is attaching to the intermediate filament, the cytoskeletal component, and these are focal adhesion spots, and that will be related to the actin group of cytoskeletal element. So, that will conclude most of the anchoring junctions in relation with a cell. Moving on, now we are going to see the other type of cell junctions, that is specialized cell junctions. That is, first one will be occluding junction or ZONULA occludens. In common term, this is known as tight junction. So, basically these tight junctions are produced by the localized sealing of the plasma membrane of adjacent cells. These are the two adjacent cell plasma membranes, and as we can see, they are very sealed closely, they are sealed very closely, and multiple proteins are included in the formation of this tight junction, and that includes occludin. We can see this color, that is the occludin protein. Next, the blue color, that is the claudin protein, and the next one is not shown in this diagram, that will be JAMs, that is junctional adhesion molecules, and that will be belonging to the immunoglobulin superfamily. And these JAMs will be present in relation with the claudins. So, this is a more clear schematic diagram showing the structure of the tight junction. We can see the 4 transmembrane domain of occludins and 4 transmembrane domain of claudins and single transmembrane domain of the junctional adhesion molecules. So, in relation with the occludins, we can see two or actually three domain proteins that are known as PDZ domain proteins. They are zonocludin 1, ZO1, ZO2 and ZO3 over here. This is ZO1, this is ZO2 and this is ZO3. So, the permeability of this tight junction which are produced by all these structures, and it depends not only on the complexity and number of strands present in it, but also it depends on the presence of functional aqueous channels formed by various Claudin molecules. So, as we have mentioned in the beginning of the video, these tight junctions determine the domains. They form the boundary between apical domain and lateral domain of a particular cell. So, that means it should allow very minimal permeability. But since the presence of Claudin molecules are there, they allow certain aqueous channels, and through these aqueous channels some of the fluids or water can pass through. To complete, the cytoskeletal element that is related with this tight junction include, that is the actin. Actin are the cytoskeletal elements over here. So, this is a schematic diagram and one portion is enlarged over here of different types of pathways of the molecule, transfer of the molecules. So, these are individual cells, and if the molecule is passing between the cell, that is known as paracellular pathway, and if the molecule is passing through the cell, that is known as transcellular pathway. What we are concerned here is about the paracellular pathway, and we have one enlarged version of the paracellular pathway over here. All these red color structures belong to the occludin group, and they form a tight tight junction between the adjacent cells, but in between them we can see multiple Claudin molecules which allow a pathway for the movement of water molecules. They form the aqueous channels within the tight junction. And where will you find these tight junctions? In the body, there are multiple regions where we can find these tight junctions in the functional aspects. Some examples include blood-brain barrier that is formed by astrocytes, blood-nerve barrier that is formed by perineurium, blood-retina barrier that is formed by retinal pigment epithelium, blood-testis barrier Sertoli cells, blood-thymus barrier that is formed by the endothelial lining, the capillary wall, macrophages and type 1 epithelio-reticular cells. You will see some diagrams. So, first one showing the blood-brain barrier. So, this cell will be the astrocyte and these are the blood vessels within the brain, and this perivascular feet will play an important role in maintaining the blood-brain barrier. These are the axons. So, the components from the blood have to cross all these structures to reach the nervous tissue, and this is the blood-brain barrier. In case of blood-retina barrier, we can see the retinal pigment cells over here, and we can clearly see the tight junctions between them over this region. So, that is the blood-retina barrier. The next one will be the blood-testis barrier. We can see the Sertoli cells very clearly over here. These are the Sertoli cells. This is one Sertoli cell. These are the processes of adjacent Sertoli cells together forming a tight junction, thereby separating the compartment into a luminal compartment and a basal compartment. This is the blood-testis barrier which is formed by the Sertoli cells. Next is blood-thymus barrier. This is the thymus and these are the blood vessels and with the RBC within it. So, outside we have the thymic tissue where developing T cells will be present, and all these structures together form the blood-thymus barrier, and the main component will be the epithelio-reticular cells present in it. So, that is the different examples of tight junctions. Moving on to the next specialized group of tight junction, that is communicating junctions. Specialized group of cell junction, that is communicating junction or gap junctions. So, this is again a schematic diagram. This is cell membranes of adjacent cells, and these green structures are the communicating junctions. And we can see each of these green structures are actually made up of a group of proteins, and each individual one is known as connexin. And through this connexins, on a particular arrangement, configuration, they will be opening a channel through which adjacent cell cytoplasm can get connected. And this is the only non-cellular structure that permits the direct passage of signaling molecules from one cell to another. And these are important, very important in tissues in which activity of adjacent cells must be coordinated. And gap junctions, mainly as we can see in the diagram, they are formed by 12 subunits of connexin protein family. How the 12 will come? Because in one cell there will be 6 connexin proteins will be there. In adjacent cell there is 6 connexin proteins, together they form 12 connexin subunits. And this entire gap junction channel consists of 12 subunits we have seen, and that together forms a 10 nanometer long cylindrical transmembrane channel, and roughly the diameter will corresponds to roughly around 2.8 nanometers. And we have seen this diagram already, and we already know some conformational changes in the connexins leading to opening or closing gap junction channels. And these functions, actually we have identified these functions with the help of atomic force microscopy. Some of the important clinical implications of these gap junctions include the mutations in connexin 26 gene will cause congenital deafness, that is due to the defective recirculation of potassium in the cochlear sensory epithelium. Other one will be the connexin 46 and 50 genes mutation, and that will be resulting in inherited cataracts, and both proteins localized within the lens of the eye. So, the coming 2 slides will describe the cell junctions in a nutshell. So, you can take screenshot of this page and you can compare different types of cell junctions, specialized cell junctions very easily. This is the first page, and this is the second page. You can compare them, just compare the classification, then major link proteins connected to it, or the cell adhesion molecules connected to it, then the extracellular proteins, then which cytoskeletal component, and sometimes look into the functions of this individual cell junctions also. So, that concludes the topic of cell junction.